Hello, IB Bio students. So today we're going to continue with 8.3, and that means that we are starting photosynthesis. We're going to be looking at the light-dependent and the light-independent reactions. But before we delve into those topics, first we're going to look at the chloroplast and the structure of the chloroplast. So the essential understanding says that the chloroplast is adapted to fit to its function in photosynthesis. So pretty much like the mitochondria, form fits function. That's the main idea that you should always remember. So what are the structures of the chloroplast that you need to learn, and what is a chloroplast? So a chloroplast is an organelle found in plants, and they convert light energy into chemical energy. So that's exactly what they do, right? The chemical energy may be either ATP for the light-dependent reactions or in the form of organic compounds in the light-independent reactions, which we'll talk about that in another video. Now, what are the tissues in plants that possess chloroplast? So it's not the whole plant that possesses chloroplast, it's only the leaves, but you're not going to find any chloroplast in the roots or even the stem of the plants. Now, just like the mitochondria, chloroplasts are thought to have been independent prokaryotes that were internalized by larger eukaryotes via endosymbiosis. So hence again, chloroplasts are good evidence for the endosymbiotic theory. They have a double membrane, just like the mitochondria, and they have their own DNA, just like the mitochondria, both circular and naked. And they have ribosomes, 70S, just like the mitochondria. And they are also, some metabolic processes are also susceptible to certain antibiotics, just like the mitochondria. So how is the chloroplast form adapted to its function? So let me scroll this over, and this is what you need to know. These are the main structures that you need to know. So let's start with the outside. So notice that there is a double membrane. There's an inner and an outer membrane, which is evidence for the endosymbiotic theory. Not much happens in this double membrane, unlike the mitochondria. We have the stroma, which is the internal fluid of the chloroplast. But something very important indeed does happen in the stroma, and that's the light-independent reaction. And within the light-independent reaction, we have the Calvin cycle, which we will talk about in other video lessons. And then we have these, they look like quarters to me, and we call these thylakoids. So these thylakoids, they're these flattened discs, and these discs have a very small volume, so it maximizes surface to area ratio. And because they have a very small volume, they have the ability to generate a hydrogen gradient, so a proton gradient or electrochemical gradient, just like we saw in the mitochondria. So with the thylakoid, there are thylakoid membranes. And within that membrane is where we see the electron transport chain, ATP synthase, and photophosphorylation happening. So it's within that membrane of the thylakoid that we see the light reaction. And within the light reaction, we find the electron transport chain, and we find the process of photophosphorylation. Now, if we were to stack a whole bunch of thylakoids, so if we were to stack a whole bunch of these discs together, then we have a granum or a grana, and that's when you get a whole bunch of them. And again, this is to increase the surface area to volume ratio, and small internal volumes quickly accumulate ions. So that's the good thing of stacking them like this. And then you can connect the separate grana via the lamella. So these are the structures that connect the grana. And that's pretty much it. What does the lamella do? It maximizes photosynthetic efficiency. So for this particular topic, you should be able to, as a skill, annotate a diagram of the chloroplast, just like you were asked to do for the mitochondria, and indicate those adaptations that help maximize the photosynthetic efficiency of the chloroplast. So on a test, you might see an electron micrograph like this, or you might just see a diagram like this, and you need to be able to understand what you're looking at. So here in this label, you see the membrane in dark purple. It's hard to see the double membrane, although it's still there. You see the grana right here, so the stacks of thylakoids right here. And then you see the lamella in lighter green, which is what connects all of the grana together. And then you can also see some starch granules that appear as dark spots. So all of these dark spots here are starch granules, but they color-coded them in light pink for you to be able to differentiate them. But these little dark spots here are also starch granules. And again, you see the same thing here, right? The stroma, which is the internal fluid cavity of the chloroplast. The thylakoids here stacked into grana, and then the lamella connecting the grana for greater efficiency. So that's it for today. Make sure you learn how to label and annotate.